start recording of the meeting. So this is Wednesday. This is our second meeting for Bio 208, again for fall 2020 semester. And by now, I ask that you work through most of chapter 19 to be at the end of the red blood cells and the end of the part of the chapter where you have gone through what's in plasma. You know what's in plasma. You know each major item and the importance and the reason that it's there. And you've gone through what is the red blood cell, where does it come from, how is it produced, what's its life cycle, what is key about its structure, what's key functionally inside of it, helps us, again, purposefully maintain homeostasis. And I kind of rushed through, but these were the questions that you should be able to answer just off the top of your head. You should be able to think about and respond with some information to these answers. The other big thing I wanted to hint is when someone pulls blood work and we look at, again, just a red blood cell pool, and we want to see for anemia purposes, or maybe for leukemia purposes, or maybe just to kind of get assessment of what does their blood look like. Usually when someone gets a blood count or a blood report, one of the things it'll show is, of course, hematocrit. Right? And remember, the hematocrit, when you initially spin blood, is that division line. And so in females, you know, it's going to be slightly lower because, one, we have a little bit less muscle mass. We don't have that testosterone forcing us to increase our muscle mass and size, requiring more actin and myosin interactions, requiring more need for ATP. So females might be in that 37 to 40, 42 range, and then the males might be more in that 40 to 50, even as high as 55 range. In general, when we talk about hematocrit, we usually say 40-60. All right, so usually 60% of your blood is plasma and 40% is the formed elements. All right, so that's usually kind of the line, just to make, again, in physiology, we love to make numbers and math simple. So 40-60 is generally what we say hematocrit's going to be. So that 40% of formed elements, and again, because most of the formed elements are going to be red blood cells, that is normally, make the leap. Hematocrit typically also represents the red blood cell count and the oxygen carrying capacity of the body. We are taking big leaps to be able to say that, because it could be you could have a 40% of your blood is formed elements, but you could still have more than normal white blood cells and less than normal red blood cells. And so again, that number is just a starting point to say, this is how many formed elements you have. So someone could be potentially 40% hematocrit, so that would be normal, but they could technically be still anemic because that 40% might actually be very low in red blood cells and high in white blood cells. Okay, so the first line that we normally look at for people is, what is the hematocrit? Then we'll look at, all right, what are the red blood cell counts? And again, it should mostly be red blood cells. And then for those red blood cells, we want to know that about 1% of your red blood cells should be lost every day. So about 1% of your red blood cells should be coming into circulation every day. And the red blood cells that are coming into circulation are not fully biconcave and have not fully gotten rid of all the RNA and all the ribosomes. So they're still churning out in these little factories that are going to, because there's RNA, so that that phosphate bond there, and those nucleotides are still there. They are going to have little purple spots. And so we are going to call those cells reticulocytes. So again, your reticulocyte number, if you can read this, it's about 0.8 to, again, 1% of your red blood cells are coming into circulation on a daily basis, and therefore you would expect your red blood cells are being lost at about that 1% rate. Okay? And so when your hematocrit is showing it's at, let's say, 37 or 38%, if you were being told, hey, you need to take iron supplements, you need to eat a little bit more protein, we want to get your red blood cell count up, you would hope that your reticulocytes would actually be a little bit higher, maybe at 1%, 1.1, 1.2% more of the red blood cells that we found in that blood sample, because then you know I'm probably adding more cells than I'm taking out. And so hopefully in a few days to weeks, my hematocrit will come up, especially if it's near that 37 or it has been below that 37%, telling us you're probably anemic. Okay. Now remember, free-floating hemoglobin is not a good thing, because that would be because your number of red blood cells that are blowing up and lysing and losing their hemoglobin to the cytoplasm, and then the cytoplasm is what we filter in the kidneys, and so that hemoglobin gets lost to your urine. We know that there's going to be a little bit of hemoglobin in there, but there shouldn't be a lot. And again, 12 to 18 grams per deciliter, that's really, really, really small amount, but you know, there should be a little bit, again, just telling us that we do have some red blood cells blowing up. All right, your red blood cell count, again, for normal purposes, should be somewhere between 4.2 to 6.3 million per microliter. That's a lot of cells in a very small, where's my mirror, small amount of fluid. And again, because of the testosterone effect, men tend to be near that 5 million and above. Women tend to be between that 4.2 to 5 million. And again, that's just a process of men have more ATP demands and higher resting basal metabolic rate, so they tend to have more red blood cells, more oxygen being carried per second per minute of the day. Okay, now these two next volumes, they were in your lab manual as something you could potentially calculate. Again, we don't do math on a lab exam. We don't do math a lot of times. So you need to know what they mean. So the MCV means the mean corpuscular volume of a red blood cell. So what this means is if I have a red blood cell, okay, and I was to stick a needle in it and I was to suction out all the fluid inside, so all that would be left would be the cell membrane and the antigens on the cell membrane and the chlorine bicarbonate exchanger protein and the phospholipids and the cholesterol that's part of the membrane. And everything inside would be pulled out. So I would be pulling out all the hemoglobin, all the water, all the sodium, potassium chloride, hydrogens, bicarbonate that's suspended in the water, all the carbonic anhydrase, all the glucose, pyruvate, and intermediates between that, and all the enzymes that help in glycolysis convert glucose to glucose 6-phosphate, glucose 6-phosphate to fructose 6-phosphate, fructose 6-phosphate to 1,6-biphospho-fructose, and all those steps of glycolysis. I don't remember all of them. But all of those molecules, if I was to suck them out with the water, and again, most of it is hemoglobin, I would get a volume that is inside the cell. And that cell's, on average, volume is known as its mean corpuscular volume. And by getting a number of what is about the inside of the red blood cell, I, in general, know how much fluid is in the red blood cell, okay? And so that's an important thing to let me know. Are my red blood cells the right size? Because they could be too small, and so the mean volume is too small. Or they could be too big, and so then the mean corpuscular volume could be too much. And if they're too small or too big, that could run into problems with oxygen carrying capacity, and then that stackability and that ability to move through the blood vessels appropriately. Now, because I want to know more specifically with red blood cells, how much of the cell is actually hemoglobin? If I know how much volume I have in the cell, how much of it is actually hemoglobin, that is known as your mean corpuscular hemoglobin concentration. So to give you an analogy, in this bottle, if this was the red blood cell and I was to drink or suction out all of the contents, that would represent the mean corpuscular volume. How much fluid is in here? And on average, it says in these are supposed to be 12 fluid ounces. Okay, but if I want to know how much of this is actually the syrup, that would be something inside this volume. There's a certain amount of syrup that makes the taste. Well, how much of the volume is that syrup is like saying, well, how much of the red blood cell is the hemoglobin That's the second one. Okay, stand by for a second. Everybody talk amongst yourself. I got to go make sure my son's on his music class. Stand by. 1 second. Okay, go to music. All right, so he wasn't at his meeting, like I said. Okay, so that's what I wanted to review before we move into today's big point, all right? All right, so today's big point is white blood cells. All right, so we've hit the formed element that's the most numerous, the most robust, and the most important in terms of cardiovascular, cardiorespiratory, delivering oxygen to the body. As we are using the system to move things around the body, your white blood cells are going to hitch a ride. And the majority of your white blood cells, their goal is not to be in the blood supply, it's to get into connective tissues. And so what's the quickest and fastest way to get from the hip bone, the long bones, the epiphysis, the sternum of your body, you know, the sternum, anywhere those flat bones are located, where the red bone marrow is located, we'll take a ride on the red blood cell with the red blood cells in the bloodstream and get to your location. All right. So all white blood cells, again, when the guy looked through the microscope, he saw that there were red cells, and then there were these other cells that their cytoplasm wasn't red. So he called them leukocytes. All right, leuco for white, cytes for cells. All right, and most of these cells are going to be immune cells in function, and they have some role in the immune system. Some of them are going to play a role in the non-specific defenses. So the defenses that it doesn't matter if it's a fungus, a virus, a bacteria, a worm, a single-celled amoeba, it's going to react the same to all pathogens. And then some of them are part of the specific immune system, and that means that they have a very unique role in helping the body target a virus or target a bacteria or target a pathogen that we know is inside of us and that we're at war with, and we're trying to kill it and remove it as quickly as possible. Okay, so those are the white blood cells. All right, some of them can live a really long time, especially memory cells. And then as we reinfect ourselves and re-expose ourselves to different pathogens, those memory cells help us, again, robustly respond and make new memory cells that then live another long time until the next interaction happens. Okay, so one of your jobs in lab and one of your jobs for lecture is, if I give you these cells by a picture, if I give you these cells by the plastic models we have in the lab, if I give you these cells by some of the characteristics in the text, you should be able to correctly identify them. Okay. Now, the original guy, when he first looked at it, he went, okay, these cells, oh, I can't make a line. All right. These cells have these bumps in their cytoplasm, so I'm going to call these granulocytes, all right? And these cells don't really have a lot of bumps in their cytoplasm, so I'm going to call them A granulocytes, okay? Now, the granulocytes are going to, again, those bumps are going to be full of vesicles, full of contents that the cell can release through exocytosis, and those molecules can either punch holes or attempt to punch holes in the plasma membrane or the cell wall if it's a plant or something else, and try to break it into pieces. Okay, some of those molecules also are what we call cytokines. And cytokines is a word, it's a cell-to-cell communication system. And some of those cytokines are going to be signaling markers to the surrounding cells to do different things. So some of the surrounding cells that are immune cells, it's going to say, hey, come here and help me. Some of the surrounding cells, like muscle cells in and around a blood vessel, it's going to be If you're damaged, constrict. If you're not damaged, dilate. Some of those signaling markers are going to be going to the bone marrow to say, hey, I just let a bunch of my cells attack an area, so I need to start replacing them. Okay. And some of those cytokines could be signaling messages to the brain to say, hey, bring the heat, turn the fever mechanism on. All right. So cytokines and some of these vesicles are going to contain a lot of different signaling molecules. Okay. And they're going to have effects locally. They're going to have effects a little bit further away, and they could have effects across the entire system of the body. All right. So we talked about this in lab yesterday. The most numerous in the blood, white blood cell, is the neutrophil. All right. And it's neutral because I guess it had a neutral looking background. It's polymorphonuclear. So that means the nucleus has divided into multiple little bulbs or beads that are kind of interconnected. So it's taken the DNA and kind of moved it into 5 or 6 or 3 or 4 different little kind of bulges, and it has lots of vesicles, and the vesicles might stain more of like a purplish blue in background. Okay. So if you don't know and you're looking at a blood smear, chances are the white blood cell you're looking at 50 to 70, I think up to 70% of the time is a neutrophil. So if you don't know a cell and you're looking at a blood smear on the microscope, if you don't know, the best guess is probably a neutrophil. All right. Eosinophils are another cell like the neutrophil that has a lot of vesicles full of material, and it's only going to have 2, so it's bilobed. All right. Back in chapter 4, neutrophils and eosinophils, we collectively called microphages, small cells that can do phagocytosis. The way they do phagocytosis on something that's bigger than themselves is they release their vesicles, and it's kind of like they spit on something to get their chemicals to start breaking it apart, to get in more of the eosinophils and neutrophils and macrophages to come over, and then they start eating it. All right. So they spit, and then they eat small pieces and parts of cells and debris. All right. So that's microphages, and that's their intracellular contents. Now, they are kind of polar opposites, and I kind of explain it as the neutrophil, let's say, is going to throw acid on molecules. So its vesicles are going to be made up of cellular molecules that are going to maybe try to break a part of cell membrane, like you were throwing acid on it. And acid's not going to kill everything. So eosinophils are going to be a different set of cytokines and a different set of cellular killing molecules that if they're released, they're like throwing base on something. They're really not acid-based. I'm just trying to use that as an example to show you that even though they had the similar spit and eat, one of them is going to try to use a type of breaking down molecules that's going to work for a lot of things, but not everything. And so the eosinophil is going to be a different type of cellular breakdown molecules that will hopefully kill and break down all the things that the neutrophils can't do. Okay, and so collectively, both of them together should break apart almost anything and everything thrown at us. Okay, basophils are going to be our most least likely to be in the bloodstream. All right. We have a ton of basophils, but the basophils like to get to their residence, which is your connective tissues in your airways, in your skin. And when they take up permanent residence in those connective tissues, we call them Mast, M-A-S-T cells. And so those mast cells, again, are the ones we associate with like allergens, all right? And so they're the ones that get sensitized if you are inhaling constantly ragweed or molds or doing different, exposures to different things that the body knows is foreign, and the mast cell somehow decides it's foreign and it's deadly and it's going to kill us, so we're going to respond to it. And again, they release their chemicals, and if you've ever had someone have an asthma attack, some of those chemicals they release to constrict the airway, are going to cause swelling, are going to cause inflammation to occur. So we know that some of those cytokines they release change muscle contraction of the airways, change muscle contraction and relaxation on blood vessels, change the accumulation of fluid, so things swell, change the temperature of the environment, so everything gets hot and causes, a big swollen, hot, injury looking site to occur. All right. And again, we call those mast cells when they live in connective tissues. They're basophils in circulation, and they are the least likely to be seen, and they're probably the most difficult to identify because, again, they're least likely to be seen. And so if you look at this picture, I can't really tell where the nucleus is and where the vesicles are. It's a little bit clearer here, but again, it's a challenge. All right. And so if you ever are looking at blood smears, chances are the white blood cells you would least likely to see would be the basophil. Okay. Which white blood cells are most affected by the AIDS virus? Okay, we'll get there. All right. Hold on. Nope, it's actually going to be T cells that are most affected by the AIDS virus. All right. Now, so those are our granulocytes. All right. Now, the other category is A in front of granule, so it's now without granules, and those are going to be the macrophages or the monocytes, and then our lymphocytes. Okay. Now, the macrophage is one of the largest white blood cells. So it's physically the biggest cell, and it has what looks like this kidney bean type nucleus shape, and then it has cytoplasm around it. Are there vesicles in the cytoplasm? Yes, but by the naked eye, there's one maybe there, one or two here. It's just not as many vesicles by the naked eye. So that was why they were put in the A granule category. But technically, they do have vesicles. All right. And monocytes are what we call these cells when they're in the circulatory system. And when they take up residence and do not then travel in the blood anymore, so whether that's in the spleen, the liver, the lungs, the airways, the skin, the connective tissues in our heart, connective tissues anywhere in our body, they are then called macrophages, and they can be fixed in a spot. So in the liver and the spleen, they might be like holding on to the little bit of connective tissue that's not going to move as blood flows. So they're fixed to try to, not move with the blood. Or they could be a little bit of free floating. And so again, when those cytokines start being released by other cells, they will, like ants, hone in and run straight to the injury site to help eat what's going on. Their mojo is I'm going to eat or phagocytose whatever I can, and I can phagocytose big cells because I'm pretty big, and then I'll worry about what I ate by mixing it with my lysosome organelle and chewing up and tearing apart what I ate inside of me. And then I'll take pieces and parts of what I ate and I'll put them on my membrane of my cell, and I'll go and present that to the T cell and say, look, I ate this. Is this good? Is this bad? Is this self? Is it not self? What did I get? What do I eat? It's like a child, like, here, look, I ate this. I ate this. What is it? What is it? Okay. So they are sometimes known as antigen presenting cells because when they eat something, they go and bring it to the T cell and say, hey, I ate this. Is it self? Is it not self? Is it bad? Is it good? Did I find something? Did I find a pathogen? Okay. The last category of cells that we can see with the naked eye are the lymphocytes. Now, lymphocytes can be natural killer cells, B cells, or T cells. And without being able to see what is on the membrane and the antigens and the proteins embedded in their membrane, I cannot tell you for sure if I'm looking at a T cell, a B cell, a natural killer cell, and if I'm looking at a CD8, a CD4, a T helper cell, or a T memory cell, a T suppressor cell, a B memory cell. I can't tell you because by the naked eye, all I can tell you is that's a lymphocyte. All right. So T cells and B cells are huge this is what we learned with AIDS. They are huge in your specific defenses. The T helper cell was the target for the AIDS virus. And when the T helper cell went away, your macrophage ate all these cold virus, flu virus, bacteria, and had nowhere to present it to say, hey, I found this in the body. Is it self? Is it not self? Is it a pathogen? And because there was no T cells there to say, yes, that's a bad thing, let's ramp up defenses, people died of pneumonia, people died of colds, people died of staph infections. All right. So we know from really the 80s, the T cells, the B cells are huge in our specific defenses, but by the naked eye, by just looking at these cells, I can't tell the difference. So by text, but for now, our purpose is this right now, by text, by visual, by model, all you can tell me is these are lymphocytes, and you can't tell me which type. To do that, you would have to be able to tell me, what is this protein? What is this protein? What is that one? What is this little thing on the membrane? And then I could potentially tell you what type of T or B, or what, if it's a natural killer cell. Okay? All right, so hopefully that helps guide you for lab. Be prepared that maybe 2 or 3 of these cells, by picture, by model, or by the text provided, and there's text also provided in your lab manual, you should be able to identify these cells. All right, and again, for our purposes right now, you don't need to be able to identify anything beyond this is a lymphocyte. Okay, all right. For white blood section, if you look, this is very similar to how we get red blood cells. All right, so in our bone marrow, we have these hematopoietic stem cells. They're also known as hemocytoblasts. Remember, blast in bone meant it's a building cell. Hematopoietic means same thing. It's a cell that's going to help give rise to our blood cells. Hemocytoblast, it gives blood. It's a cell that's building blood. Okay. Now, that cell, when it goes through mitosis, it produces one daughter cell that replaces dad and becomes the resident stem cell. The other daughter cell is going to have one of 2 options. It can become what's known as a myeloid cell, or it can become a lymphoid progenitor cell. Right? Myeloid is for multi. So if it's going to become the myeloid cell, that is how we can still create red blood cells, we can create platelets, or we can create any of our granulocytes or our monocytes. Okay? How does it know to go this route? Well, there's a lot of factors. And if you go back and look, and I don't think I put it here, no. If you go back and look at the slides earlier in the chapter, there are a lot of cytokines. And some of the cytokines are interleukins, so they're abbreviated IL. Some are these colony stimulating factors, CSFs. Okay? And then there's hormones like EPO, for erythropoietin hormone, and TPO, which is for thrombocyte potent hormone, thrombopoietin hormone. All right, those also play a role. So this little cocktail of signaling molecules interact with the daughter cell and can push it to become the myeloid or push it to become the lymphocyte producing stem cell. Okay, and when you start to push it towards the myeloid, that's just one step. Eventually, again, EPO, TPO, other interleukins, other colony stimulating factors then start to tell that myeloid progenitor cell to become what we then will generate a red blood cell, so that erythroblast, or a prothroblast, or another still myeloid type cell that can become what will eventually lead to one of our 3 granulocytes or one of our monocytes. If we knew the perfect cocktail to get red blood cells, and I mentioned this on Monday, I could harvest from all of you, maybe from your hip, I could harvest a bunch of hematopoietin cells, and if I knew the perfect cocktail of colony stimulating factor and how much, and interleukins and how much, and those other factors, and then how much EPO, I could grow the perfect amount of red blood cells in fluid, and I could then replace having to do blood donations. I don't know the perfect cocktail. I don't know the perfect amounts. Every time they try to do this, they always end up with some cells not becoming red blood cells and not becoming really maybe fully functional. So that's why we still need people to donate blood, because we haven't figured this out 100%. But we know colony stimulating factors are key to get that stem cell to become a myeloid. And then we know colony stimulating factors, some interleukins, some other cytokines, and EPO will give us red blood cells. Colony stimulating factors, interleukins, and TPO will give us platelets. And again, certain interleukins, certain colony stimulating factors will give us one of the other granulocytes or a monocyte. Okay, lack of colony stimulating factors, lack of EPO, TPO, and other interleukins and other types of cytokines could tell that daughter cell to become our lymphoid progenitor cell. And if that happens, then we can end up with a T cell, a B cell, or a natural killer cell. And again, I don't have the perfect solution or cocktail there to do this outside of the body. So it's more complicated, like everything in the body. It's a little more complicated than just this perfect little map drawn here with you need multi-CSF, you need EPO. If I just needed that, I could do this outside the body. I don't just need that. There are some other little fine tuning, small amounts of this and that. I don't know all of them. So that's why I can't make this without, I can't give blood to people made exclusively in the lab without being made from the body. Okay. So the point to take away is all of your blood cells come from one source, your bone marrow. And that's part of the reason why if someone has bone marrow issues, you could be affecting, yes, the white blood cells and immune system, but you could also be affecting the circulatory system and the red blood cells and their platelet ability, their clotting ability because of platelets. We know that for sickle cell, somehow the genes, when you get into the pre-thromboblast and the actual make a biconcave shape, one of the genes more in this pathway is affected. So someone with sickle cell, all of this is fine, but somewhere a gene in this region of the pathway is where the cell is not creating the right genetic physiological push to make the cell look eventually like this when it matures. Okay. If you have a gene somewhere, let's say in this route, that could be where you end up with a problem just with basophils. So you end up with a type of leukemia and maybe even a lymphoma because basophils, mast cells show up in your connective tissues. That's specifically a basophil-derived cancer. Okay. So this is part of the reason, again, sometimes if this cell is the problem, you have issues across all of here. If the problem becomes more of the genes in one of these more narrow areas, you might have okay red blood cells, okay platelets, but you have a problem just specifically to the neutrophils or the macrophages or the monocytes, one of those cells. All right. Okay, so in summary, after today, hopefully you have a better understanding of red blood cells versus white blood cells. How do we differentiate into them? What are the influencers? And you can start to identify those white blood cells by either picture, model, and text. Okay, again, we talked about this at the beginning of class. On Thursday, I'm trying to do the Zoom for a live stream of lab. I'm not opening it up to everybody yet. So it's only for 60A, 60B folks because I have 2 or 3 people that are going to attend this. The 2 people who didn't come to lab on Tuesday and my 1 person who can't come to lab because they work with COVID patients. So the health form would always say, don't come. They are going to come to lab on Thursday through Zoom. And if it works, and if it goes okay, we're going to try then to offer that up every Thursday for all of you in the online sections. If your schedule allows, you can attend the live labs. And then you can see what they're doing. Then when you're doing the ADILT labs, it might make more sense why I made you do that activity that way. And then as you work through your lab manual, it might make sense. It might also help you reconnect with friends so you can meet some of the people that are maybe attending the live lectures and the live stream labs, and you see people in lab that you recognize their names. So you can reach out to them maybe through email or in the chat and try to, again, connect with friends that y'all can do a virtual study group, or maybe y'all can try to meet with the tutor or meet with, go to the bone lab at Forest Park together, okay? So be patient. The other thing I'm trying to do tomorrow is I'm going to try to have a Microsoft Teams session with my 207 class, and if that works, I'll try to set up to maybe have a virtual office hour where you can join me on Teams on Thursday mornings. I still have about, I think it was like 17 people have not accepted the email invite to ADI. So please look in your junk, look in your spam folder for STLCC emails or your my.stlcc.edu email and find that invitation. I resent it to everybody today. You need to have your ADI accounts ready for the weekend because you can start doing your heart labs this weekend if you want. Next thing is, again, Friday's lecture, we're going to go over the last part of this chapter is just the clotting. So I want to hit, again, clotting by platelets and the intrinsic, extrinsic pathways, and then continue to work on your own, watching, listening, viewing these videos on YouTube already posted. If you're tired of listening to me, you want to hear different stories, you want to hear different analogies, Anatomy GMC, she's from Tennessee, she has a drawl, but she does a really good job. She would be someone. You can also see if Jeff Baumstark's videos are available if you want to listen to him, if you want to hear a guy's perspective. You can even try and see if Kelly Roberts has some of her AMP2 videos and lectures available. Those are other Merrimack campus people that I know use YouTube for videos. All right, so you don't have to listen to me if you get bored of my voice. I'm okay with that. All right, continue to work through the lab manual and try to get through the chapter 19 blood lab by the end of the week. Make sure you fill out Dora's survey. Again, if you need to, oh, I totally spelled Sally's name in the chat wrong. It's S-K-N-O-P-P-E, not S-K-L. But contact her. There are 2 tutors for A&P on the Merrimack campus, and then contact S. Roussin, R-O-U-S-S-I-N 2 at mystl.edu. My.stlcc.edu, because she is your contact for the bone at Forest Park and the tutors at Forest Park. Okay, and I don't know who they are. I don't know that person, so hopefully they'll be able to give some information. And then remember, the quiz is Sunday. The key of this quiz is to do Respondus. Hopefully everybody has a plan on how to try to take this. If you do not have a Chromebook that accepts the Respondus, or an Apple computer or an iPad, please try to find a solution. We need to have a backup plan, because if we all go virtual, everybody's taking labs and everybody's taking lecture tests online, and you have to have this system set up. Okay, I'm going to stop the recording.